Hello everyone. In this lecture, I will talk about polymerase chain reaction, which is one of the most important cytogenetic techniques that you must learn in this course. In this lecture, you will know some basic information about PCR as well as the various types of this essential technique. Polymerase chain reaction, or PCR, is an important cytogenetic technique used to make millions and even billions of copies of a defined region of DNA in vitro. This technique was developed by Kary Mullis in 1985, and this invention was the reason why he received a Nobel Prize in the 1990s. Please remember that the amplified products after polymerase chain reaction are called amplicons. The success of PCR depends on a number of factors, with its reaction components playing critical roles in amplification. So I will first discuss to you the different important components needed in PCR. The first and most important component of PCR is the template DNA strand, which acts as your sample. A PCR template for replication can be of any DNA source, such as genomic DNA, complementary DNA synthesized by reverse transcription from an initial RNA sample, and lastly, your plasmid DNA. Next is DNA polymerase. DNA polymerases are critical players in replicating the target DNA. DNA polymerase is an enzyme that makes new strands of DNA, like DNA replication in an organism, using existing strands as templates. Amplification is usually carried out by Taq polymerase, which is a thermostable DNA polymerase I enzyme from Thermus aquaticus. The thermostability of Taq polymerase is an essential requirement in PCR, as PCR reaction will go through different temperature stages that may denature or disable the non-thermostable polymerase. Taq DNA polymerase has a relatively high thermostability with a half-life of approximately 40 minutes at 95 degrees Celsius. The next component is the PCR primer. So any region of any DNA molecule can actually be chosen to be amplified. The only requirement is that the sequences at the ends of that region are known. The oligonucleotides, which act as primers for the DNA synthesis reactions, define the amplified region. PCR primers are synthetic DNA oligonucleotides of approximately 15 to 30 bases. PCR primers are designed to bind via sequence complementarity to sequences that flank the region of interest in the template DNA. This figure shows why PCR primers are important. So when the primers are bound to the template strand, they can be extended by the polymerase, and then the region that lies between them will get copied. Another important component is the deoxynucleoside triphosphates, or DNTPs. Your DNTPs consist of 4 basic nucleotides: cytosine, guanine, thymine, and adenine. These DNTPs are added to the PCR reaction in equimolar amounts for optimal base incorporation. The next important component of the reaction are the magnesium ions. So magnesium ion functions as a cofactor for activity of DNA polymerases by enabling incorporation of DNTPs during polymerization. In addition, magnesium ions facilitate the formation of the complex between the primers and DNA templates by stabilizing negative charges on their phosphate backbones. Lastly, we have the buffer, which provides a suitable chemical environment for the activity of DNA polymerase. Normally, PCR occurs in an environment maintained at a pH range of 8.0 to 9.5, which is often stabilized by tris hydrochloric acid. For Taq polymerase, potassium ions, which are oftentimes from potassium chloride, promote the binding of the primer to the template strand. Now that you have learned about the major components of the PCR reaction, let me explain to you the three main steps of PCR. In a typical PCR experiment, the target DNA is mixed with Taq polymerase, the two oligonucleotide primers, and a supply of dNTPs. The amount of target DNA can be very small because PCR is extremely sensitive and will work with just a single starting molecule. The reaction is started by heating the mixture to 94 degrees Celsius. At this temperature, the hydrogen bonds that hold together the two polynucleotides of a double helix are broken and the target DNA becomes denatured into single-stranded molecules. The temperature is then decreased to about 50 to 60 degrees Celsius, which is just below the annealing temperature. This results in some rejoining or binding of the single strands of the target DNA, but also allows the primers to attach to their complementary sequences. And lastly, we have extension, or otherwise known as elongation. So during this step, the temperature is raised again to about 72 to 74 degrees Celsius. And in this step, a new strand is synthesized by the enzyme Taq polymerase. This slide shows the overview of the different steps in PCR. The DNA strand is first denatured to become single-stranded molecules, then the primers anneal or bind to the ends of the single-strand DNA molecule through complementary base pairing. Then, the thermostable Taq polymerase elongates or extends the new strand until two new double-stranded molecules of DNA are formed at the end of the first cycle. In the succeeding cycles, the number of correct amplification products accumulates in an exponential fashion, that is, they double during each cycle until one of the components of the reaction becomes depleted or until the end of a defined cycle. Now, let me give you the different types of PCR. So these are the most commonly used PCR formats used in most molecular biology laboratories. Please take note of the significant features of each type. Reverse transcriptase PCR, or RT-PCR, was developed to amplify RNA targets. In this process, complementary DNA, or cDNA, is first produced from RNA targets by reverse transcription, and then the cDNA is amplified by polymerase chain reaction. The reverse transcription is accomplished through the action of the enzyme reverse transcriptase. Real-time PCR, or quantitative PCR, abbreviated as qPCR, is the technology for the quantification of nucleic acids. This method is a way of quantifying the amplification of a DNA as it occurs, which means that it detects the accumulation of amplicon after each thermal cycle in real time. Real time implies that data collection and analysis occur as the reaction proceeds. The initial concentration of target sequence can be determined by a comparison with the amplification kinetics between the target and an internal control with known copy number. The next type of PCR that I will talk about is nested PCR. Nested PCR was developed to increase both the sensitivity and specificity of PCR. This technique uses two pairs of amplification primers and two rounds of PCR. Typically, one primer pair is used in the first round of the amplification of PCR of 15 to 30 cycles. The products of the first round of amplification are then subjected to a second round of amplification using the second set of primers. The second set of primers anneal or bind to a sequence internal to the sequence amplified by the first primer set. The increased sensitivity arises from the high total cycle number, and the increased specificity arises from the annealing of the second primer set to sequences produced by the first round. Then we have multiplex PCR. So in multiplex PCR, two or more primer sets designed for amplification of different targets are included in the same PCR reaction. Using this technique, more than one target sequence in a clinical specimen can be amplified in a single tube. As an extension to the practical use of PCR, this technique can save time and effort. The primers used in multiplex reactions must be selected carefully to have similar annealing temperatures and must not be complementary to each other. PCR exponentially amplifies nucleic acids, and the number of amplification cycles and the amount of amplicon allows the quantification of the starting quantity of targeted nucleic acid. However, many factors complicate this calculation, often creating uncertainties and inaccuracies, particularly when the starting concentration of the genetic material is low. Digital PCR overcomes these difficulties by transforming the exponential data from conventional PCR to digital signals that simply indicate whether or not amplification occurred. Digital PCR is accomplished by capturing or isolating each individual nucleic acid molecule present in a sample within many chambers, zones, or regions that are able to localize and concentrate the amplification product to detectable levels. After PCR quantification, a count of the areas containing PCR product is a direct measure of the absolute quantity of nucleic acid in the sample. This is the end of my discussion about PCR. You will learn more about this topic in detail next semester in your Molecular Biology and Diagnostics course. Thank you for listening.